IB Bio, Human Physiology, Part 13 is the third of 3 parts covering neurons and synapses. In Human Physiology, Part 13, Neurons and Synapses C, this movie, the focus will be on synapses. The essential idea is neurons transmit the message, synapses modulate the message. Here is the first of 2 slides that provide an outline of all available movies for Topic 6, Human Physiology for SL and HL students. Use the outline to find the movie you need for review. This movie is focused here. Here is the second slide with the outline for all available movies of Topic 6, Human Physiology. Before looking at synapses, let's do a bit of review. Here's an IB syllabus statement that I covered in the first movie, Neurons and Synapses A. You should be able to annotate a diagram of the structure of a neuron, including dendrites, cell body with nucleus, elongated axon, myelin sheath, nodes of Ranvier, and motor end plates. This will be important in this movie. In this diagram of a motor neuron, you can see the dendrites, the cell body with the nucleus, the elongated axon with its myelin sheath, the nodes of Ranvier, and the axon terminals where the neuron would synapse with another neuron or synapse with an effector, which would be a muscle or gland. The propagation of the impulse along the membrane occurs in this direction, jumping from node to node to node, known as saltatory conduction, and the impulse moves very rapidly in this fashion. In the second movie on neurons and synapses, I covered these 2 statements. Explain how a nerve impulse passes along an axon, including the roles of sodium and potassium ions in an action potential. Include details on ion channels and active transport. Secondly, analyze oscilloscope traces showing resting potentials and action potentials. In this diagram, we can see a neuron with an expanded section of the axon. Voltmeters can be used to measure the charge differences across the membrane. Notice the charge difference, a high potential energy condition. This neuron is in a resting state with more positive charge outside the membrane relative to the inside, where the charge is more negative due to the presence of negatively charged proteins in the cytoplasm. An action potential is a wave of depolarization that propagates along the membrane of a neuron. At any location along the membrane, the depolarization is followed by repolarization. Upon the stimulus, the voltage-regulated gates open and sodium ions flow into the neuron. The membrane potential begins to shift, and if the potential reaches threshold, the action potential occurs as the potential reaches positive 30. In other words, the inside of the cell membrane becomes positive. The change in the charge milieu around the voltage-regulated potassium gates allows the potassium gates to open, and potassium diffuses out of the neuron, resulting in repolarization. The action potential propagates along the neuron membrane as a result of local currents that cause each successive part of the axon to reach threshold potential. The sodium-potassium pump reestablishes resting potential, all of this in fractions of a second. So let's turn our attention to synapses. Synapses are junctions between 2 neurons, or a junction between a neuron and an effector. The effector would be a muscle or a gland. In this diagram, the synapse between this neuron and this neuron is right here, and it's expanded, as you can see here. You can see the axon terminal of the presynaptic neuron and the dendrite, which will have receptors on the postsynaptic neuron. The impulse will propagate from this neuron, the presynaptic neuron, to this one, the postsynaptic neuron. And that's because the axon terminal of the presynaptic neuron will release neurotransmitters, as you can see here. And they release those neurotransmitters into the synapse, the gap that lies between the 2 cells. and the postsynaptic membrane has protein receptors for the neurotransmitter. Here are the two IB syllabus statements that drive this movie. Define synapse, explain the principles of synaptic transmission as exemplified by the neuromuscular junction, including the release, diffusion, and binding of neurotransmitter and depolarization of the postsynaptic membrane. Here is a diagram displaying the synapse as a junction between two neurons, 1 and 2. The synapse is the gap that lies between neurons, and it modulates the message or the signal. What that means is that the synapse serves to regulate how messages move within the nervous system. More on that later. In this diagram, you can see the presynaptic neuron that has vesicles containing neurotransmitter. The postsynaptic neuron has receptors for the neurotransmitter. When the impulse arrives at the axon terminal, the neurotransmitters are released into the synapse. They diffuse across to bind to the receptors, stimulating the action potential on the postsynaptic neuron membrane. Again, we can see the presynaptic axon terminal with vesicles containing neurotransmitter that gets released into the synapse by exocytosis. The postsynaptic membrane has receptors for the neurotransmitter. The neurotransmitter simply diffuses across the synapse. The distance is short. If enough neurotransmitter is released into the synapse, the membrane of the postsynaptic neuron will reach threshold potential, and an action potential will propagate along the postsynaptic membrane. The signal will have moved from one neuron to the next. Here is a micrograph displaying the synapse. We can identify the presynaptic neuron by the presence of vesicles ready to secrete the neurotransmitter into the synapse. The membrane of the postsynaptic neuron is right here. The postsynaptic membrane has receptors to which the neurotransmitter will bind. Here is another micrograph of the synapse. Can you identify the presynaptic neuron and the postsynaptic neuron? Here is a nice black line drawing of the synapse. There are many things here to notice. The impulse has arrived at the presynaptic axon terminal. Notice that this region of the axon terminal is depolarized, while this region has already repolarized as the action potential has moved in this direction. The depolarization at the axon terminal results in the release of the neurotransmitter from the vesicles into the synapse by exocytosis. The neurotransmitters diffuse across the synapse and bind to protein receptors on the membrane of the postsynaptic neuron and cause the postsynaptic membrane to depolarize. Notice here. You can see the region of depolarization essentially extends from the presynaptic neuron to the postsynaptic neuron. Now remember, the synapse modulates the signal. The quantity of neurotransmitter must be sufficient to cause the postsynaptic membrane to depolarize above threshold. That's the key. If the depolarization of the postsynaptic membrane reaches threshold, then an action potential propagates along the postsynaptic neuron. If the depolarization of the postsynaptic membrane is not enough to cause an action potential, then there is no transfer of an impulse. And this is how the synapse modulates the signal. Here's one more diagram of a synapse. You can identify the presynaptic neuron by the presence of Golgi vesicles, which are ready to secrete the neurotransmitter into the synapse itself. The postsynaptic membrane has receptors that will bind the neurotransmitter, causing the postsynaptic membrane to depolarize. Now, if the original stimulus is not strong enough to cause substantial amounts of neurotransmitter to be released into that synapse, then the postsynaptic membrane may not reach threshold potential. Thus, no action potential in the postsynaptic neuron. It is in this way that synapses modulate messages. Maybe the stimulus was something that did not require attention. Should animals respond to the wind or any blade of grass that touches them as they move through the habitat? So, if this is the synapse region here, and this is the neurotransmitter diffuses across the synapse and binds to the protein receptor on the postsynaptic membrane. This causes a conformational shape change in the receptor. You can see that here. And this allows sodium to diffuse through into the cell of the postsynaptic membrane. This is depolarization. The influx of sodium due to this open gate, a gate opened by the presence of the neurotransmitter, is the mechanism by which the postsynaptic membrane depolarizes. But in order for an action potential to propagate on the postsynaptic membrane, the threshold potential must be reached. The action potential arrives at the presynaptic axon terminal. Neurotransmitters are released into the synapse. The neurotransmitters diffuse across the synapse and bind with receptors on the postsynaptic membrane. The postsynaptic membrane depolarizes as sodium ions diffuse into the postsynaptic cell. The signal is sent to the next neuron if the threshold potential is reached. Now, it's important to consider what I'm about to say next. The neurotransmitter cannot remain in the synapse. Otherwise, the postsynaptic membrane would be perpetually depolarized. Thus, the neurotransmitter must be broken down by an enzyme, represented here as a black line, and the components of the neurotransmitter are reabsorbed by the presynaptic cell so that new neurotransmitter can be synthesized. Now, here are two IB syllabus statements, and the first one you've seen before, but I've modified it to include the term motor end plate. This is the junction of nerve with muscle. Muscle is an effector. Annotate a diagram of the structure of a neuron, including dendrites, cell body with nucleus, elongated axon, myelin sheath, nodes of Ranvier, and motor end plates. Now, the second statement here is very important. Explain the principles of synaptic transmission as exemplified by the neuromuscular junction, including the release, diffusion, and binding of neurotransmitter and the depolarization of the postsynaptic membrane. In the neuromuscular junction, the postsynaptic membrane is the membrane of a muscle cell. Here is a neuromuscular junction. We have the neuron here, and we have the muscle cell here. The muscle cell is exemplified by these proteinaceous myofibrils, and the term sarcomere is used here. You may or may not understand muscle cell structure, and in the IB syllabus, muscle structure is strictly a part of higher level material. So for the purposes of understanding the neuromuscular junction, just pick up on this pattern that you can see here as an indication of a muscle cell. Now, the neuron can be seen with familiar structures. Here's the axon, here's the myelin sheath, here's the presynaptic membrane with neurotransmitters ready to be released into the synapse. The neurotransmitter at the neuromuscular junction is a molecule called acetylcholine. Now practice this because you need to get to know this neurotransmitter. Acetylcholine, acetylcholine, acetylcholine. Acetylcholine diffuses across the synapse to stimulate the muscle cell to contract. Synapses with acetylcholine as the neurotransmitter are known as cholinergic synapses. Acetylcholine is the neurotransmitter at neuromuscular junctions. The acetyl group comes from the mitochondrion. You might remember acetyl-CoA. And choline, that part of the molecule, comes from your diet. Now, the enzyme that breaks acetylcholine in the synapse is acetylcholinesterase. It's the enzyme that breaks down acetylcholine in the cholinergic synapse. Now, the choline then is reabsorbed by the presynaptic neuron and reused to synthesize more acetylcholine. Now, what's the name of that enzyme again? Acetylcholinesterase. You've seen this image before, but imagine that this is a neuromuscular junction. The synapse is a cholinergic synapse. Acetylcholine is released from the presynaptic neuron to stimulate the muscle cell to contract. Acetylcholine is removed from the synapse by acetylcholinesterase, represented in this image as these black lines. The choline is reabsorbed by the presynaptic neuron to be used again. Here is another neuromuscular junction. Recognize the muscle cell by the myofibril pattern seen here. This is a cholinergic synapse here, where acetylcholine is the neurotransmitter. In this image, we can see a muscle cell here. We can see the protein myofibrils in cross-section here. And we have a neuron that has come into contact with the membrane of the muscle cell. And as the action potential arrives to the axon terminal of the presynaptic neuron, acetylcholine is released into the synapse. You can see the acetylcholine abbreviated here, ACH. Acetylcholine diffuses across the synapse, binds to a receptor on the postsynaptic membrane, which is the muscle cell. This results in the diffusion of sodium into the muscle cell, causing the membrane of the muscle cell to depolarize. The action potential propagates along the muscle cell membrane. Acetylcholine would be broken down by acetylcholinesterase. This would happen in the synapse so that it does not continually stimulate the muscle cell to contract. Choline is reabsorbed into the presynaptic neuron for reuse. And that brings us to our last IB syllabus statement of the movie. Here is an application of the cholinergic synapse. Apply the functioning of a synapse to the use of neonicotinoid pesticides used in the control of insects. Neonicotinoid pesticides block synaptic transmission at cholinergic synapses by binding to acetylcholine receptors. Neonicotinoid pesticides and organophosphates interfere with the synaptic transmission, the neuromuscular synapse, in insects. This serves to kill the insects. Use of pesticides, such as these neonicotinoid pesticides, is justified by the users of the insecticides to protect their crops, to protect the food supply for people. Neonicotinoid pesticides block synaptic transmission at cholinergic synapses by binding to the acetylcholine receptors on the postsynaptic membrane. These receptors can be seen here. If the receptor is blocked by the pesticide, then the neurotransmitter, acetylcholine, cannot propagate the impulse to the muscle cell. The insect suffers a state of paralysis, an inability to move. Neonicotinoid pesticides block synaptic transmission at cholinergic synapses by binding to acetylcholine receptors. If the receptor is blocked, then the neurotransmitter acetylcholine cannot propagate the impulse to the muscle cell. The insect suffers a state of paralysis, an inability to move. Neonicotinoid pesticides block the acetylcholine receptors on the postsynaptic membrane of a neuromuscular junction. This causes the death of the insect due to an inability to move. Organophosphates block acetylcholinesterase so that acetylcholine is not broken down. The insect dies due to excessive spasms of muscle contraction. There is concern worldwide that the use of insecticides in agriculture is having unintended consequences. Bees, insects useful as pollinators for most of the world's crops, are significantly reduced in number worldwide. The cause of bee death is being intensively investigated. One possible cause of bee death is neonicotinoid pesticides. And that brings us to the end of IB Biology, Topic 6, Human Physiology, Part 13, Neurons and Synapses, C.